So our next speaker is Qirong Lin from the Karolinska Institutet, and he will talk to us about spatial transcriptomics of B-cell and T-cell receptors that reveal lymphocyte clonal dynamics.
Thank you so much. So hi, everyone. I'm Qirong Lin, and I'm a third-year PhD student at Jonas Frisen's lab at Karolinska Institute. And it's really my honor on behalf of our team to present our recent work on spatial profile T and B cell receptors in human tissue. So before I jump into the slides, I just like disclosure that I'm a scientific consultant for 10x Genomics. I'm holding the patent right to the spatial technology I will present here. So without further, I think I don't need to emphasize the importance of our immune system to the audience, to everybody. And it helps us to maintain the homeostasis, pathogen defense, inflammation like regulation, and so on. And the spatial distribution of the B and T cells also played a very important roles for their development and their function. For example, those like activated the B cells will entering the germinal centers and go through like proliferations, have somatic mutations, and affinity selections. And besides that, we have tumor-infiltrated B and T cell that might sit next to the tumor populations. And sometimes they will form very sophisticated tertiary lymphocytes. And also they are heavily involved in infections, vaccine, and other aspects. So what if we want to study the topic of like the clonality of B and T cells within the structure or within the local area? We might have the methods about like single cell RNA-seq that a lot of people like you already know, that we can have the gene expression profile together with B and T cell receptors, but we need to disassociate the cells, which means we will lose the spatial contact of where the T and B cell is. So how about we use spatial method? For example, we have another approaches that is recently developed, spatial transcriptomics. It is conceptually similar to the single cell method, but use a DNA. It's kind of like a DNA array with a lot of spots, like 5,000 of them. And they will have the oligo to capture the mRNA and do reverse transcription and have cDNA amplifications. But the limitation of this approach is that we will fragment the cDNA library into short parts, and we only sequence the 3' end of this library, which means we will lose most of the VDJ sequence, and we cannot call the clone from the spatial. So our method, spatial VDJ, take one step further beyond the traditional spatial transcriptomics. That we took the cDNA library after the reverse transcription and amplification, and use targets enrichment like from the bees to enrich T and B cell receptors. And thanks for the introduction of the long read sequencing from the previous speaker, that I don't need to go deep into that part, but we do the full the long read sequencing to those target captured T and B cell receptors. We also developed the method which allow us to use the short the sameness PCR to enrich T and B cell receptors and allow us to do the sequencing on the normal next generation sequencing like Illumina platform as well. But we will not extend too much on this part because of the limitation. So from these steps, we are able to have the IgTR clonal informations together with a gene expression from the same cDNA library, so we don't need to prepare another adjacent section for that, but from the same cDNA and project them together with the HNase DNA. So here comes the first exciting question. So how many clones do we detect it? So we took the human tonsil and have 6 sections, and we prepared the Vision libraries, and we did the spatial VDJ experiment and to do the sequencing. And because those individual spots might have multiple cells, so the clone we defined here is not at the paired receptor, but individual receptors. But out of 6 sections, we have more than 62,000 of Ig and TR clones in total. and a lot of BCRs and the TCRs slightly fewer, especially for the TR alpha due to the technical challenge. But we recovered those clones from all the 6 sections. So each dot is from 1 section. So we have like the total number of the clone, and how does it look like on spatial? Then we can plot them back. So for example, this IgHM clone 30, they are enriched in 1 follicle, although they have some spread out spot as well. And for some of the TR clones, they are widely spread like in the tonsil area that is mostly like outside or at the border of the follicles. So those are 2 examples, but how reproducible is spatial VDJ? Because we have 6 sections. Are they just like the best out of the 6? So if we plot them on all the 6 sections, we can see such patterns are quite consistent in those in those sections, and although they will have some like spread out cells spots. And for the TR beta, they are they are outside the follicle area or at the border of the follicle area and across all the 6 sections. So from those 2 parts, we have the confidence to say, okay, we captured quite a lot of the Ig and TR clones, and they have some spatial patterns. There we can have some very interesting biological question. The first is, since we are looking at the tonsil, so what's the clonal diversities in a human B cell follicle or germinal centers? So let's revisit the human tonsil again from our data set. So based on the gene expression and the morphology, we annotate more than 30 follicles on spatial. And because the B cells will have somatic hypermutations, and they will happen on the CDR3 region as well. So we use the Immcantation pipeline to help us. We use the defined clone to group the clones with similar CDR3 sequence into the clonal family. And we use Shazam and Alakazam, like other tools and pipelines for us to the clonal family analysis and the lineage analysis. So how many clonal families do we have from the spatial? And we are mainly focusing on the IgH here because they will have a higher somatic hypermutations from the data set. And we have the follicle 1, which is rich of all the IgH clone family, and the rest of them have like hundreds of them, like someone have higher, someone is lower. And if we change the angle of view, instead of at the follicle level, but the clone family level, how does individual family looks like across all the follicles, whether they are enriched or restricted in 1 follicle, or they are spatially expanded into multiple follicles. So we have most of the clonal family are quite restricted, but we have some of the family that is quite expanded that attract our attention, that it might bring up the possibility to view any potential dynamics within the family. So we have another question. Can spatial VDJ be used to decode B cell clonal evolution in space in that clonal family? So out of all the clonal family, I just want to pick up the 2 of our favorite one. So one is IgHM clone family, and they have a very extensive hypermutation and form a very complicated linear tree with different branches and different subtrees inside of it. And on the contrast, we have another tree, which is IgHA already had the class switching recombinations. And it looks like they have like it has 2 major somatic hypermutation or expansion event, like one is over here and another one is in the bottom. Then how does this tree looks like on spatial? Like how do those spots comes from? So we project, since we have 6 sections, we project all the 6 the spot from 6 sections into 1. So we will find some like spot looks like they are outside the tissue, but they are actually from other sides. They are still within the tissue, but we are trying to like merge them together. So most of the spots are located inside the dominant follicles, the follicle one, the biggest one. But still, we have some of the spots looks outside of them, or even entering the like other follicles. Then we are very curious about that, like whether those spots are enriched in one subtree and have another branch that is spread out outside, or they have another patterns. So if we count the UMI count of each subtree that we separate them, and to count the proportion of the reads within either follicle 1, the dominant one, or other follicles, or in the extrafollicular area, we find most of them, like the dominant proportion of the UMI, are within the follicle 1. Like those 3 have the dominant one over here, and some of reads are spread into other follicles, and some expand into extrafollicular area. But there's 1 tree over here, are mainly extrafollicular. So that was very interesting to us. How about another tree, like looks completely quite different from the previous one? So if we group the tree, the nodes from different parts, so we have 2 parts. One is on the top, we color them in the dark green, and another one we color them in the blue, and to plot them on spatial. And surprisingly, they are distributed in different follicles. Like the dark green one is located in 1 follicle, and the blue one are mainly enriched in another follicle. And interestingly, we have this dot. It looks like a blue dot next to a dark green population. It's actually the root of the bottom tree. Another one here, 69, is this dot. It's more associated with the previous lineage instead of this one. So this is like a very exciting discovery for us. So we also did like other research. For example, we extend our method to a breast cancer patient, and we found the tumor adjacent, or like those clone that is close to the tumor area. And we developed the tools to, because since it's a spatial method, it's not at the single cell level, but we can use their spatial patterns to pair the light chain with the heavy chain. It's called repair. And we also have other method, like for example, we want to have a ground truth of what we have, what clones we have. Then we developed, it's called bulk SMART63, that we can enrich all the clones, and we can have a reference to compare with them. But as a roadmap for the spatial VDJ, we can provide multiple B and T cell clones at the same time with the spatial context from a leftover cDNA library, or which means you don't need to prepare the library from the very beginning, from the sample. And also TMB cell clones have reproducible spatial patterns. And since it has a long read sequencing, it allows us to do a linear tree or B cell clonal evolution study. So with that, I want to thank all the amazing collaborators and co-authors listed here, especially some shout out to Camilla and Long and Kim Thring, which have a tremendous works on experiment and analysis, and also Jeff Mold, who did a lot of experiment work and a very good idea, and Hossein as initialized the long reads analyze, Alma and Shong, who developed the repair method to overlap to find the paired light chain and heavy chain on spatial, and Xin Song as an expert in the breast cancer help us with the breast cancer related topics, and all the other co-authors and funding agencies, and feel free to scan for the paper, and I'm welcome for any questions. Thank you.
Thank you very much, Qirong, for this very interesting talk. We have a couple of questions in the chat. Well, first one that I was also wondering about, if you did observe any co-localization of also clonally related T cells with these clonally related B cells that are parked to different germinal centers.
So you mean like, is there any associated T cell, like B and T cell population with overlap spatial patterns? Is that what you mean?
Exactly, like if any like co-localization with T cells that are also clonally related across these two different germinal centers that you have just shown before.
That's a very good question. So one of the limitations, or actually the fact that we encountered in contrast to the single cell, is like we might have a lot of T cells, but from our experience, we will have like less B cell from the single cell RNA-seq. But for the spatial, since plasma cells and B cell will have way high expression on their receptor compared to the T cell, so we are captured like fewer UMIs on the T cells and might be diluted by the surrounding like the B cell and other cells. So we have T cell clones, but it's not significant enough for us to confidently say they are spatial overlap. And we are still like improving the method for like capturing of both T and B cells, especially T cells.
Okay, yeah, thank you very much for the answer. Maybe a second question, I think we still have time. If you could use the clonal lineage information to build a sort of spatial pseudotime.
Oh, and that'll be very, very interesting. So besides the data I present here, we also have gene expression, and also we use a tool developed also by Alma and Xiong. It's called a stereoscope, which can deconvolute the cell type proportion on the spot. And with that, we haven't like really stepped into it, but we can have the mask of what the cell type we have on spatial together with their gene expression. And such a linear tree can allow us to like mount the gene expression profile along the branches and to see how it looks like. But there's one limitation is those each individual nodes, maybe I can move back. Yeah, for example, those nodes here might have reads from different spots, and they might have different composition of the cell, the predicted cell types, which can mix up the gene expression pattern. So I'm not 100% sure what's the best way, but maybe we can based on their like gene expression, or let's say single cell data sets, and to see how to link them together. Yeah, from the spatial itself, it's hard to say because it's kind of like a mixture of the cell type, but we can think more about it. Yeah.
Okay, thank you very much. Very interesting technology, and we are looking forward to further developments here and integration with single cell, as you have just mentioned.
Yeah.
Maybe for the sake of time, we also can move now to the next speaker, but there's plenty of questions in the chat, which we encourage you to answer.